Ladies and gentlemen, I would like to thank the organizers for giving me the opportunity to present our study SARS-CoV-2 seronegative subjects target CTL epitopes in the SARS-CoV-2 nucleoprotein cross-reactive to common cold coronaviruses. I do not have any conflicts of interest to disclose. It has been observed that there is a great variety in the course of SARS-CoV-2 infection regarding the severity. However, the reasons for that are not yet clearly defined. It is known that both antibodies as well as T cells play a critical role, and SARS-CoV-2 reactive T cells have been found in seronegative donors. This led us to our hypothesis that infection with common cold coronaviruses induces SARS-CoV-2 cross-reactive T cells. First off, we blasted for homology stretches between SARS-CoV-2 and the common cold coronaviruses OC43 and HKU1. We were able to identify a 15 amino acid homology stretch in the nucleocapsid that we termed DP15. To test for cross-reactive T cells in vitro with this DP15 peptide, we recruited a total of 139 donors, consisting of 23 healthy donors and 116 HIV patients on ART treatment. They had a median CD4 count of 692 per microliter and a median viral load of below 20. After isolating the PBMCs, we stimulated them with the peptide DP15 in the presence of IL-2. After 10 to 14 days, outgrowing cells were either tested in interferon gamma ELISpot assays or via intracellular cytokine staining for both interferon gamma and CD107A. Using those peptide stimulations assays, we found that 48 of our 139 donors responded either to DP15 or shorter peptides in its sequence, so equaling a responder ratio of almost 35%. Importantly, all those responders were seronegative for SARS-CoV-2, indicating that they have not been infected with the virus yet. In our cohort of healthy donors, 4 out of 23 people, so about 17%, responded. In our HIV-infected cohort, the responder ratio was almost 38%. The median ELISpot frequency in stimulated T cells was 48.5 spot-forming units, with a range of 10 to 493 SFUs. To characterize those DP15 reactive T cells in further detail, we then did intracellular cytokine staining for 11 responders. And here we were able to show that interferon gamma was secreted by both CD4 as well as CD8 T cells in response to DP15. Furthermore, 7 out of 11 CD8 positive T cells also expressed CD107A, marking them as cytolytic cells. As we were able to show that also CD8 positive T cells secreted interferon gamma in response to DP15, we wanted to map down the HLA class 1 epitopes in DP15 further. Therefore, we did truncation experiments with shorter peptides to define the N and C terminal borders of our epitopes. Then we did HLA restriction for those epitopes using HLA type B lymphoid cell lines. We found a number of epitopes within DP15. For 3 of those, we were not able to get the HLA restriction yet, but for the others, we found that the epitope DY10 is restricted to A02, the epitope LY9 is restricted to A29, and the epitope SL9 is restricted to both B7 as well as B35. When looking at the HLA types of our responders and non-responders, we found that at least 50% of A29 and B7 positive subjects recognized either DP15 or shorter peptides. However, it has to be stated that there's actually a higher frequency of B7 and B35 in our study cohort compared to the general population due to a biased recruitment, as we did HLA binding predictions of our peptides beforehand. Since we saw that most of the B7 positive subjects in our study cohort were responders, we wanted to have a closer look at the corresponding epitope SL9. SL9 also has an homologous peptide in OC43 called LL9 with an S to L substitution in the first position. When testing SL9 reactive T cells with both peptides, we found that they responded equally well to SL9 as well as LL9. And we could also show this in peptide titration assays. Furthermore, in FACS-based killing assays, we could see that SL9-specific T cells were able to lyse peptide-loaded target cells, which clearly shows that they are cytolytic effector cells and also stresses the importance of this epitope. To be able to analyze a larger proportion of our cohort for cross-reactivity to common cold coronaviruses, we used the peptide DL11, which encompasses several of our identified epitopes. The homologous peptide in OC43 or HKO1 is called QL11 with a D2Q and an S2L substitution. And we found that actually QL11 was significantly better recognized by DL11 reactive T cells, which demonstrates the in vitro cross-reactivity and also indicates an induction of T cells by the common cold coronaviruses. In conclusion, we could show that SARS-CoV-2 specific T cells can be found in SARS-CoV-2 seronegative subjects. Both CD4 as well as CD8 T cells were able to recognize epitopes within our peptide DP15. And furthermore, we were able to identify several new SARS-CoV-2 OC43 cross-reactive epitopes. Taken together, this indicates that infection with common cold coronaviruses induces a T cell response that is then at least in part cross-reactive to SARS-CoV-2. As for our HIV-infected subjects, we were actually able to see a twofold higher recognition compared to the healthy donor cohort. This can be partly explained by biased recruitment of specific HLA types. Compared to the healthy donor cohort, most of the HIV-infected subjects were typed for HLA class 1. Furthermore, there's also actually a higher percentage of CD8 positive T cells in HIV-infected subjects. They had a median CD4 to CD8 ratio of 1.1 versus 1.9 in healthy donors. Furthermore, the persistent immunoactivation that is observed in HIV could actually also lower the trigger threshold for the outgrowth of peptide-specific T cells in our stimulations assays. But still, this higher frequency of SARS-CoV-2 reactive T cells could also be a possible reason for why treated HIV infection does not seem to be a strong risk factor for the development of severe COVID-19. But taken together, prospective studies are necessary to assess the functionality of cross-reactive T cells. I would like to thank all my colleagues from the University Hospital Erlangen as well as the Friedrich-Alexander University Erlangen-Nürnberg that worked with me on this project, but especially my two supervisors, Professor Thomas Harrer and Dr. Christa Ganu. Our research work has been funded by the German Research Foundation, DFG, the COVIN project, and also the Hector Foundation. Thank you very much for your attention.